So, what is genetic engineering? Genetic engineering is when the genetic code of an organism is altered to produce a desired trait. This is done by taking a gene from one and inserting it into the DNA of another to get the wanted end result.
Showing you 5 foods that have been genetically modified beyond recognition. Number 5. Carrots. Carrots have been around for thousands of years. In fact, seeds dating back to 5,000 years have been discovered in Europe. However, originally carrots looked a lot different. They were small and white rather than very long and orange. The carrots we have today are known as Western carrots. However, some other carrots still exist, which are most commonly called Eastern carrots. These carrots can be smaller in size and also even purple rather than orange. Number 4. Bananas. Bananas are believed to have been first cultivated around 7,000 to 10,000 years ago. The long yellow banana most people know are a result of centuries of careful breeding. They descended from 2 wild banana species called Musa acuminata and Musa balbisiana. These 2 banana species don't taste like bananas we have today. In fact, one has lots of hard seeds in the middle. However, thousands of years ago, banana cultivators discovered that cross-pollinating these 2 plants produced a sweet, yellow seedless fruit that tasted really good. This is how modern-day bananas were born. Number 3. Watermelons. Experts agree that watermelons originated somewhere in Africa and spread to the Mediterranean. One of the watermelon's earliest ancestors was first cultivated in Egypt around 4,000 years ago. This watermelon was hard, bitter and pale green in color, however they did still contain lots of water. It's believed the watermelon was an excellent source of water for people, especially during the dry seasons, and that's the reason why they grew them. It's also believed it was the Egyptians who began to selectively breed the sweetest and best watermelons over many years, which led to the tasty sweet watermelons we have today. Number 2. Corn. It's believed corn is one of the first crops ever cultivated by humans around 10,000 years ago. However, at that time, corn was a lot different to what we have today. The earliest ancestor of corn that we know of is something called teosinte. This plant doesn't look anything like corn today, but they did produce kernels which were a lot smaller. However, on a genetic level, the plants are almost identical. Over thousands of years of artificial selection, corn has changed massively in its appearance. This was done by people selecting only the plants that produce the most kernels. Number 1. Peaches. Fossils of peaches have been discovered in China that date back over 2.5 million years ago. However, these peaches would have been a lot smaller than today and resemble small cherries. Once again, through artificial selection, the peaches that were the biggest and contained the most flesh were chosen. This led to the peach being much larger with lots of juicy, edible flesh. It's believed it only took about 3,000 years for the peach to turn from a small cherry-like fruit to what it is today.
To get a better sense of how this is done, let's first understand how DNA works. In any living thing, the DNA acts as the instruction manual for the body. Certain genes code for certain proteins, which in turn make up all the different parts of the organism. Any trait that you can think of is coded by DNA. Now, once you know what codes for what, you can use genetic engineering to essentially edit the parts you want, to some extent. all living things use the same genetic code, DNA, made from the same 4 bases, so scientists can use special enzymes to cut parts of the DNA from one living thing and sort of sew them onto the DNA of the target. The result is then a living thing that has a new gene and a new feature as a result of that gene. Now, there are many examples where genetic engineering is used today, but we're going to focus on a very specific medical example to understand this concept better. Humulin, a medication used for diabetics, is created with genetically modified bacteria. Humulin can be given to diabetic patients who lack the insulin they need. Now, how they do this is scientists figured out which section of our DNA codes for insulin, and they also found certain enzymes that can cut that gene out. What they do is, using enzymes and heat, they isolate a specific gene and then insert it into the DNA of bacteria. The result is now a bacteria that produces human insulin. And since bacteria can multiply really fast, you can create a lot of insulin in a very short amount of time. Now, this is great! Genetic engineering poses huge potential in the field of science, providing us with new insight on how our genes work.